Okay, this is podcast number 1 for standard 3.5. In this standard, we're going to be taking a look at basic genetics, basically looking at Mendel's laws of segregation and independent assortment, and we're going to be going over a lot of vocabulary within this section. So for this standard, you will be able to identify the law of segregation. You'll explain the experiment used by Mendel in order to come up with this law of segregation, and that's specifically called the monohybrid cross. You'll identify what the law of independent assortment is, and you'll describe independent assortment using something called a dihybrid cross. Okay, so a lot of really important vocabulary that we're going to concentrate on within this particular podcast, and then we'll get more into the experiments in further podcasts from here. All right, so genetics in general. Genetics is the study of heredity. So when we talk about heredity or an inheritance, it's something that gets passed down from parent to offspring. And so in genetics, that we're talking about is the actual genes or the makeup that makes you who you are being passed down from parents to offspring. These characteristics end up being your traits, what you look like. Okay, so all of your characteristics that you have been determined by the genes that you have received from your parents. So that could be your hair color, your height, skin color, anything that is how you look or what your body does very often ends up being a genetic thing. So if somebody who is lactose intolerant, that's actually a gene that would cause that to happen, and that's something that would have been passed down from parent to offspring. So when we study heredity, we're studying how traits get passed between generations. And a lot of times we can look at some of the different things that can happen along the way and why certain genes or traits are shown more than others. That's with dominant and recessive traits. Sometimes you get a mixing effect with certain types of genes. So we're going to kind of explore each of those different types of heredity that can happen as we go through our genetics unit. Okay, so heredity again is passing traits on from parent to offspring. And remember, we just learned that a parent or an offspring is never identical to a parent because offspring are a combination of two different parents. So they have some genes from both parents, and so they end up being a combination of the two. But genetics will explain why sometimes you might look more like one parent than another. All right, so Gregor Mendel is considered to be the father of genetics. He was an Austrian monk in the mid-1800s. He was also the gardener for the monastery where he worked, and he was a high school science teacher. And one of the things that he did in his garden is he actually worked with pea plants. He would grow pea plants on a regular basis. That was part of what he harvested for food for the rest of the monks. And as he was working on these pea plants, he started to notice some characteristics about them, and he started to get curious. So when he was working with his pea plants, what he actually noticed is that there are 7 distinct traits that he could easily see within pea plants. Things like the height of the plant, flower position, so where the flower came off of the stem, the color of the flower, the seed and pod color, and the seed shape and the pod shape. So these are things that he could actually definitely see. And what he found was that for each of these different traits, there were 2 forms that he could easily see. For example, flowers were either they were either purple or they were white. So for every single one of these traits, there was always 2 different forms that showed up in the pea plants. And so he started to get really curious about that. Okay, so this just kind of show you what some of those different characteristics are. So seed shape, it's either round or it's wrinkled. The color, yellow or green. Purple or white for the flower color. So everything had one of 2 possible characteristics that you would see when you were looking at the particular pea plant. And so he really got curious about that because he's like, well, there's only 2 different versions. I wonder what this means. And that's when he started to do some experiments. Before we start going into experiments, though, we're going to first go over some of the essential vocabulary that you have to know in order to be successful with a genetics unit. Okay, so we're going to start with genes. The traits that Mendel saw, what he was actually looking at, were caused by pea plant genes. Now, at the time that Mendel was doing his work in the mid-1800s, nobody really knew anything about DNA or genes. He just knew that there was something that was causing these characteristics to show up. Now, we now know that a gene is actually a piece of DNA, and that piece of DNA is going to determine the protein that the cell will make. And that protein, in turn, is what is going to decide what you look like. Okay, so your genes determine what you look like by deciding the proteins that your cells make. So if you have brown eyes, that means that the cells that make up your eye are producing a protein that is making them brown. So that's kind of how the genes work, is that they're going to tell your cells to make certain proteins, and that's going to give you the traits that you have. We will go into more detail on how DNA does this later in the unit. Okay, so remember, your chromosomes contain many genes. We've talked about chromosomes and how they have different genes all along them. They're a segment of the DNA within there. And remember, you have 23 pairs of chromosomes, and all of them have genes on them. And so you end up with thousands and thousands of genes that make up the human genome. And each gene is going to code for a different protein and tell your body to do a different thing. So as we're talking about genetics, what we're really talking about is how chromosomes are being passed from one generation to the next, because on those chromosomes are the genes. All right, so getting into some of the essential vocabulary. Alleles describes a gene that has two different forms. So when we just talked about Mendel's pea plants, there's an allele for purple flowers and there's an allele for white flowers. So we call those two different forms of the gene alleles. Okay, another example is with the height gene. There's two alleles for height, tall and short. Okay, and when we talk about alleles, an allele can be either dominant or recessive at this point. There are exceptions to this rule that we will go over later, but just to keep things simple, we're going to start with the alleles as being either dominant or being recessive. If an allele is dominant, it will always show when present. So if you have a dominant version of the gene, you are going to show whatever trait that dominant allele is for. To show a recessive, you have to have two copies of it, because a recessive will be hidden by a dominant allele. So if there's a dominant there, it's going to show. So the only way to show a recessive is to have gotten a recessive from your mom and a recessive from your dad. And that will give you your recessive trait. Okay. Now when we talk about alleles, we actually use symbols, so that when we do genetic crosses like Punnett squares, we can make it a lot easier for ourselves. Usually the symbol that we use is the first letter of the dominant trait is going to be used in either upper or lower case. If it's the dominant, it'll be in upper, and if you're talking about the recessive allele, it'll be in lower. Okay, so using our height gene again, tall is dominant, so we're going to use a capital T to represent the tall allele. The recessive of tall is short, so we're going to use a lowercase t to represent the short allele. We do not use an S. We're going to use the same letter so that we know we're talking about the same gene, but we just use capital for dominant and we use lowercase for recessive. So you wouldn't use an S for short. You're using the first letter of the dominant trait. Okay, so in this case that's tall, so we're using T. Now occasionally when we're doing this, sometimes the letters aren't the easiest ones to use, because actually like the letter S can be kind of hard, because it's maybe hard to tell the difference between a capital and a lowercase s. So there are exceptions to this, but in general this is what we talk about. You take the first letter of the dominant trait, and that's the letter that you're going to use when coming up with your symbols for your alleles. Okay, now for every gene, remember, an organism has two copies. And those copies are on your homologous chromosomes. So remember we talked about homologous chromosomes are your matching pairs of chromosomes. You got one from mom and you got one from dad. And we said that they were matching because they had the same kinds of genes on them, but they were not identical to each other. Okay, so those, you could have two copies of a gene, those are your alleles. It might not be exact copies. They might be a little bit different. Okay, so if you have a tall pea plant, it could have two dominant alleles. We call that big T, big T. And that means it got one big T from mom and one big T from dad. Or it could have one big T and one little t. And that means it got the big T from one parent and the little t from the other. But it's still going to be tall, because tall dominates over the short. So it still looks tall. So these two plants would actually look the same. Now a short pea plant can only have two recessive alleles. So short would be little t, little t. The only time that you get the recessive is if there's no dominant present within that combination. And that's how you can show the recessive trait. All right, so what we're talking about when we talk about these short plants or the tall ones, sometimes when we've got organisms that have genes that are the same, we call that homozygous. Another word for homozygous is purebred or true breeding. And basically what it means is that when your homologous chromosomes come together, the genes or the alleles that you have on those chromosomes are the same type. Okay, so in the case of our height gene, big T, that's homozygous. Little t is homozygous. And remember, we've learned that the word homo means same. Okay, and when we're talking about using the word zygous is actually referring to the zygote. So in the zygote, when the two versions of the chromosomes come together from mom and dad, the two copies of that particular gene are the same. They're both either dominant or they're both recessive. Okay, so this picture is showing you, here we have one parent that was big Y, and here's the other parent that was big Y, and so the offspring that they make is big Y, and that's homozygous. Okay, the opposite of this is heterozygous. Heterozygous, another word for that is hybrid. You can think of like a hybrid car, which uses both gas and electricity for powering the motor. So hybrid is kind of like a mix. And this is where when the genes that come together are going to be different. They have different alleles. So remember, the prefix hetero means different. So again, if you look at how this works, you have your different and zygote again, that should be a thing, is zygous is zygote. So you have a different zygote. So that means when you are making your offspring, the two alleles that come together are not identical to each other. Okay, in the case of a heterozygous individual, it is the dominant allele that will be displayed. So in this case, big T, little t would be tall, but this is a heterozygous individual because they have both a dominant and a recessive. But because the dominant's there, that is what they're going to show. Okay, so that's what heterozygous is. Now, when we are talking about these letters that we're using, what we're actually referring to is the genotype. A genotype is the genetic makeup of an individual. It is literally the genes that they possess, and we're going to represent those genes by the letters that we've been using to represent that particular trait. Okay, so big T, little t, and little t, all of those things are genotypes because they tell you the alleles that are present. So they're telling you what the actual genes are that the individual has. So if the genotype is big T, that means it's got two tall genes. Okay, so genotype is just the letters that you have that represents the genes that you have. Okay, in contrast to that is the phenotype. The phenotype is the physical characteristic, and a lot of times I'll remember pheno and physical both start with PH. So this is basically what an individual is going to look like. That is your phenotype. So in this case, like big T, and big T, little t are tall. So the phenotype is tall. Okay, whereas in little t, the phenotype is short. So remember, if you look at this picture, a phenotype is determined by its genotype. So here you have a genotype, and it's going to code for a particular phenotype, what it looks like. The genotype are the genes that are on the actual chromosome, and then the phenotype is what you have when you get the end result of what the individual looks like.